रिवाइज कीजिए 6.9 ह्यूमन जीनोम प्रोजेक्ट को। In the preceding section, you have learned that it is the sequence of bases in DNA that determines the genetic information of a given organism. दूसरे वर्ड में जेनेटिक makeup of an organism or an individual lies in the dash dash sequences. If two individuals differ, then their dash dash sequences should also be different, at least at some places. These assumptions lead to the quest of finding out the complete sequence of human genome. With the establishment of dash dash techniques where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining dash dash sequences, a very ambitious project of sequencing human genome has launched in the year dash dash. Human Genome Project, that is HGP, was called dash dash project. You can imagine the magnitude and the requirements for the project if we simply define the aims of the project as follows. बताइए ह्यूमन जीनोम में लगभग कितने बेस पेयर्स होते हैं? Human genome is said to have approximately 3 into 10 to the power 9 base pair, and if the cost of sequencing required is 3 US per base pair, यानी कि the estimated cost in the beginning, the total estimated cost of the human project would be approximately 9 billion US dollar. Further, if the obtained sequences were to be stored in typed form in books, and if each page of the book contained 1000 letters and each book contained 1000 pages, then 3300 such books would be required to store the information of DNA sequence from a simple human cell. The enormous amount of data expected to be generated also necessitated the use of dash dash computational devices for data storage and retrieval and analysis. HGP has closely associated with the rapid development of new area in biology called dash dash. What were the goals of HGP? बताइए. First point from first line of NCERT. Second point. Third point. Fourth point. Fifth point. Sixth point. The Human Genome Project was 13-year project coordinated by the Department of Energy and National Institute of Health. During the early years of the the Wellcome Trust UK became a major partner. Additional contribution come from dash dash comma dash and others. The project was completed in knowledge about the effect of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent the of disorders that affect human beings. Besides providing clues to understanding human biology, learning about non-human organisms sequences can lead to an understanding of other natural capabilities that can be applied toward solving challenges in healthcare, agriculture, energy production, environmental remediation. Many model organisms such as bacteria, yeast, a free living non-pathogenic nematodes, that is fruit fly, plants that is rice and etc. have also been sequenced. चलिए इसके मेथडोलॉजीज को जल्दी से रिवाइज कीजिए. The methods involved. इस मेथड में इन्वॉल्व है two major approaches. One approach focused on identify all the that are expressed as referred to as that is ESTS. ESTS का फुल फॉर्म बताइए. The other took the blind approach of simply sequencing the whole set of that contained all the coding and non-coding sequence and later assigning different regions in the sequence with a term referred to as dash. For sequencing that the total DNA from a cell is isolated and converted into random fragments of relatively sizes. Recall, DNA is a very long polymer and there are technical limitations in sequencing very long pieces of and cloned in suitable host using specialized vectors. The cloning resulted into of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used were bacteria and yeast, and the Vectors were called the back या बास। फुल फॉर्म बताइए बीएसी का। फुल फॉर्म बताइए वाईएसी का। The fragments were sequenced during automated dash dash that worked on the principle of a method developed by dash dash. Remember, dash dash is also credited to developing method for determination of amino acid sequences in proteins. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping dash dash for sequencing. Alignment of these sequences were humanly not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently dash dash and were assigned to each chromosome. The sequence of chromosome 1 was completed in only in May dash dash. This was the last of the 24 human chromosomes 22 dash dash and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on a dash dash and some repetitive DNA sequences known as dash dash. One of the applications of polymerization in repetitive DNA sequences that shall be explained in a next section of DNA fingerprinting. माइंड कैप्चर कीजिए फिगर 6.15 ए रिप्रेजेंटेटिव डायग्राम ऑफ ह्यूमन जीनोम प्रोजेक्ट। बताइए कंप्यूटर के मॉनिटर स्क्रीन पर क्या क्या लिखा गया है।